so today we're going to focus in on these receptor interactions, right? B cells have antibodies on their surface. Do you guys remember which one of the 5 classes of antibody will be found on their surface? It's also the first class of antibody we produce. So it's IG and then a letter. Is it A, E, M, or G? M. M. IgM is the B cell receptor, right? So this antibody is its receptor. And then when it secretes this, in what form is that M? It's really mega big, right? It's 5 antibodies hooked together. We call that a pentamer. But on the surface of the B cell, it has its antibody. That's its receptor. And each B cell has its own unique antibody, right? We talked about that last time, how they literally rearranged their DNA to make a unique antibody, all unique unto themselves. Same thing happens for T cells. But remember, we have 2 main types of T cells. We have helper T cells and cytotoxic T cells. And that determines what cells they talk to. So notice that T cells actually communicate with cells. And they do this via their CD marker. So each one of our T cells has its own CD marker. Helper T cells have CD4. And I always remember 4-H, right? That's a group that teaches children about taking care of animals and stuff like that. But 4 for CD4, H for helper in this case, right? So if you remember 4-H, remember CD4, right? You remember that that's on the helper T cells. And unfortunately, that's the marker, that's what the HIV virus grabs onto and how it gets into our helper T cells. So that's why it's such a bad virus, because it's targeting our immune system, right? Cells that we need for our immune system to function properly. The other type of T cells are CD8 T cells. These are your cytotoxic T cells that will actually kill other cells. So I remember 8, the number 8 almost sounds like eat, right? And these cells can essentially eat or kill, destroy other cells. So that's how over the years I've, my little play on words, how I keep them straight, CD4 versus CD8. In addition to that, this marker allows them to only talk to cells displaying the other receptor that matches the CD4 or CD8 receptor. So this cell is presenting antigen, right? And it's presenting it using a receptor called MHC. We have MHC class 1 and MHC class 2. MHC class 1 is found on all of your nucleated cells. So any one of your cells that gets infected, it can present antigen on its surface. So which one of the T cells do you think we want to be able to interact with your cells that are potentially infected? Do we want a helper T cell or a cytotoxic T cell? Cytotoxic, because we want to kill that cell that's infected, right? So cytotoxic T cells have CD what? 8, right? Remember 8, eat. Helper T cells don't kill. Helper, as the name implies, help other cells. They don't directly hurt cells. So, remember we have some cells that can present antigen, right? Do you guys remember which ones are going to gobble up and then break up pieces and present antigen? Do you remember who can do that? Who can phagocytose and then go one step further and present that information? Macrophages. Neutrophils can phagocytose, but they don't antigen present. Dendritic cells, right? And then one more that we haven't talked about, B cells, when they interact with antigen, will also then present it as well. Okay, so your major antigen presenting cells that are going to talk to helper T cells, CD4 T cells, are your macrophages, your dendritic cells, and your B cells. So when your T cells are going through the maturation process, and what organ does that happen? Thymus. That's why they're named T cells, right? So this video that I asked you guys to look at ahead of time, I don't know why that's doing that. It's really annoying. Animations, videos. I even put some stars next to it, right, to help you guys key into it.
T-cell development takes place within the thymus, a specialized organ where T-cell progenitors mature.
That's to plug this in.
The thymus.
T-cell progenitors enter the thymus through high endothelial venules.
I'm going to start it over.
T-cell development takes place within the thymus, a specialized organ where T-cell progenitors mature and differentiate. The thymus is divided into several different anatomical locations, and as thymocytes mature, they pass from one environment to another. T-cell progenitors enter the thymus through high endothelial venules and migrate to the subcapsular region, where these thymocytes start the process of maturation by rearrangement of their T-cell receptor genes. As maturation proceeds, and the cells begin to express a specific receptor, they move further into the thymic cortex. Interactions between the T-cell receptor and MHC peptide complexes displayed by the thymic cortical epithelial cells play an important part in the fate of the thymocyte. At this point, the developing thymocytes express both CD4 and CD8, as well as an alpha-beta T-cell receptor. A cell that has a receptor able to recognize an MHC class I molecule receives both a survival signal and a maturation signal. Eventually, the cell stops expressing CD4 and maintains expression of CD8. Thymocytes that obtain a survival signal are said to undergo positive selection. A cell that has a receptor able to recognize an MHC class II molecule also receives a survival signal and a different maturation signal. Eventually, this cell stops expressing CD8 and maintains expression of CD4. Thymocytes whose receptors are unable to recognize either MHC class I or MHC class II molecules fail to receive any survival signals and die by programmed cell death or apoptosis. Thymocytes that are able to recognize MHC class I or MHC class II peptide complexes too avidly receive a strong signal that drives them into cell death. In this way, thymocytes capable of responding to self-peptide antigens are eliminated in a process known as negative selection or central tolerance. The surviving T cells migrate from the thymic cortex to the medulla. At this stage, they remain capable of recognizing self-peptide antigens expressed on other cell types, such as dendritic cells or thymic macrophages, and receive a sufficient signal to cause them to undergo programmed cell death. The remaining thymocytes, now mature naive CD4 and CD8 T cells, pass out of the thymus, either returning to the bloodstream directly, passing into venules, or via the lymphatic system.
Right, so a little bit more complicated system for them because we have two different main types of T cells. And even within those types, before this thing starts going on me, close it out. Okay. And I don't know what that beeping noise is on my computer. I cannot figure out what was going on, but it thankfully went away. Sorry if it was distracting. So they got to go through that process of checking out those receptors, making sure, right, they're communicating properly before they're released from the thymus. So they'll either be selected to move forward or they'll be killed. So as we looked at, where did I want to go? Last time. There we go. So when we talk about that process, right, that's up here. That's forming that naive cell, right? So in this case, this is B cells, right, with antibodies as their receptors. But the same thing goes true, right? Now you have a naive, they've never seen antigen before. Now from that process on, it's referred to as clonal selection, when one actually interacts with an antigen. So for T cells, it's a little bit more complicated, and sometimes for B cells too. But the important process that's always going to happen is if they recognize and they get the correct signals, they're going to become activated. Once they activate, they proliferate. And in the case of B cells, for sure, too, they're going to differentiate into a cell type we know as plasma cells. These are going to actually secrete the antibodies that they're programmed to make. And then you're going to get also the production of memory cells. These are long-lived cells that'll stay around for our next exposure. The same is true for helper T cells and cytotoxic T cells. When they're activated, you're going to get memory cells produced. So we go from naive, right, haven't seen antigen, but they do have their receptors, to being activated once they interact with the antigen, which is going to cause them to proliferate, right? That's clonal expansion. You've got copies of that same cell. So you have, instead of just one soldier, you've got many soldiers that are all the same and have the same ability. So what roles do these guys play? So one thing that wasn't shown there is that helper T cells are sometimes important in that process of B cell activation. That B cell going to proliferation, differentiating into plasma cells, and producing memory cells. What's the difference between the primary and secondary responses? So what's the difference between the first time we see something and the second time we see it? Yep. The second time, the major benefit you have is that usually you're going to be activating memory cells instead of naive B and T cells. Now, some antigens, in order to make antibodies, you need T cell help. You need the helper T cell. So those would be T-dependent antigens. T-independent antigens obviously do not require the help of the T cell. No helper T cell needed. So T-dependent and I'm going to actually play that animation today. So I'm going to go to that one. So T-dependent antigens. So this is when you need the T cell help in this process.
Antigens, such as bacteria or viruses, are usually ingested and degraded into small fragments by phagocytes, such as macrophages. The small fragments are then complexed with a special type of protein called a class II major histocompatibility complex protein, MHC, and transferred to the surface of the phagocytic cell. Helper T cells recognize an antigen when it is presented on a class II MHC because helper T cells have special recognition proteins on their surfaces called T cell receptors. This interaction activates the helper T cells. The activated helper T cell is now capable of stimulating B cells to become antibody-producing plasma cells.
And what's not shown there? Memory cells were also produced in that process, right? So in this process, helper T cells are activated by that macrophage. That T cell would also go through a proliferation process, right, where you'd have a whole bunch of them that recognize that specific antigen. Notice again the T cell receptor and that other marker, that was CD4, that allows them to communicate directly with MHC class II, right? So 2 and 4 go together. MHC class I only interacts with cytotoxic T cells, CD8, right? So there's a specific, almost like special handshake between these cells. So they know how to communicate with each other, and they know what they need to do after passing on that information. So that's an extra step, right, in the process of B cell activation. So notice here in this diagram, right, we went B cell, it went right to activation. It would happen here at this step of activation that the helper T cell would communicate with the B cell. And notice again, the B cell will actually present the antigen on MHC class 2 to be able to communicate with that helper T cell. So that's when you need T, where this picture you could say is T independent, right? No T cell involvement. And it depends on the type of antigen, on whether the B cells can go through this process on their own without the helper T cell.
So MHC2 is only on B cells?
MHC class 2 is only on antigen presenting cells, which a B cell can present antigen. What else? Dendritic cells and macrophages. Yep. So this is just showing you, right, that the B cell, once it interacts with the antigen, it is going to take it in internally. And it is going to present it on an MHC class 2. And that will allow it to communicate with that helper T cell. Right, that can help in the activation. Right, that's T-dependent antigens. The important thing is each time a B cell is activated, whether it's with the help of a T cell or not, you're going to get the production of antibody. Remember, the first antibody we're going to produce is IgM for the most part. Commonly, we're going to switch to IgG. Remember, what's one of the benefits of IgG? Why do we like that antibody? Why do we produce a lot of it? What can it do? IgG. Huh? Well, all antibodies are going to help fight infections, right? What's the difference between IgG and IgM? IgM is mega big, right? So that one's limited to the bloodstream, where IgG is just that monomer. It can travel across the placenta even. And it is a much longer lived antibody. Stays around longer. And remember with the outcomes that we've looked at, opsonization, cross-linking, it can do all the outcomes, including antibody-dependent cellular cytotoxicity by communicating with natural killer cells. It can cause neutralization. It can cause immobilization. It can activate complement. It can do everything, y'all. Everything. Whereas some of the other ones are limited, right? IgA, we just put into our secretions, right? We're only going to make that in areas where we're making secretion, whether it be mucus or tears or saliva or breast milk. So that's the very first time we're exposed to antigen. And notice there's a gap here between exposure to antigen and when the antibodies are produced. This lag period between exposure and antibody production, here it says 10 to 12, the average about 7 to 14 days. So this is true when you get vaccinated, right? There is going to be a lag period before your body actually produces antibodies or cytotoxic T cells or both. It's got to go through that process of finding a naive cell that actually recognizes the antigen, whether it be a B cell or a helper T cell or a cytotoxic T cell or all three. We've got to find the perfect match. Or matches, right? Because as it comes to antigens, they're quite complex, right? We'll make antibodies against lots of different pieces of that antigen, right? Against different epitopes. That's where specifically the antibody binds. But still, that's going to take time. Once we have that first activation, we are going to get the production of memory cells, right? Of memory B cells and T cells. So who gets activated upon second exposure? Is it naive B cells or is it memory cells? Memory cells, right? So notice no lag time, right? Because this is the second time we've seen it. We remember it. We respond that much faster and aggressively, right? Notice how quickly the antibody concentration goes up and how quickly we switch from M to G. That more protective class of antibody that can do more things. Right. So our response is faster, swifter, because we're activating memory cells instead of trying to find a naive cell that's never seen antigen before. So when you get vaccinated, right, that's probably your primary exposure, right? If you get exposed again, then that's why your response is going to be so swift and so protective, because we're activating memory cells. And you might even already have antibodies still around that can go into activation right away. So plasma cells, right, are a differentiated B cell, an activated B cell, right? As I said, they're going to increase their rough endoplasmic reticulum because it's rough because it has what on its surface? Ribosomes. Because remember, what are antibodies? They're proteins mostly that are secreted, and the rough ER makes secreted proteins. So these cells get really big. Under the microscope, the usual staining that we do, you can't tell a B cell from a T cell or a natural killer cell. But plasma cells, if they happen to be in your blood, which is rare, they would have a really huge cytoplasm. And so these guys are pumping out antibodies, millions per second. Once the infection is cleared, they're going to undergo apoptosis, programmed cell death. No inflammation, right? But as long as that antigen is still present, they're going to still keep producing antibody. And so the titer, the concentration increases as we go throughout infection. So one of the things that I did when I worked in research is the armadillos that we would infect with leprosy, because it won't grow in artificial culture, right? So they would purposely infect armadillos to grow the mycobacteria in order to study it. We would take blood samples from the armadillos during infection, and their titer, their concentration of antibody against that would go up, right, as they progressed through the infection, right, until they got to the point where we would harvest their organs to be able to get the bacteria back out. We would harvest their spleen. We would harvest their lymph nodes. The lymph nodes would get huge with the infection, right? Your spleen, remember, filters your blood, right? So it would grow in their spleen. And they would have lepromatous leprosy, which means that they only produced antibodies, right? They wouldn't have a T cell response, so they wouldn't kill off the bacteria. Unlike people, we obviously wouldn't give them drugs to kill the bacteria we're trying to grow it in that case. Their liver, too, would get chock full of the bacteria. So each B cell, we said, makes their own unique antibody. In the process, once they become activated, when they are copying themselves, they are going to purposely mutate their DNA that makes this B cell receptor ever so slightly. And the hope is they'll become even stronger binding between the antibody and the antigen. So they have a way of detecting how well they're binding with the antigen. So in this case, for demonstration purposes, they're putting a ++. That gives you an idea of how strong that binding is. Where this mutation that happened, it had a little bit less binding. It's only a 1+. So this B cell, because it's not making a very strong interaction with the antigen, it won't be signaled to proliferate. So notice its lineage dies. Where this one has the strongest affinity at that point, so it'll be given the signal to continue to proliferate. But it's still going to mutate its DNA ever so slightly in that region of its antibody receptor, its B cell receptor. So you'll see that it got lucky here. This one actually has a much stronger affinity, 3 pluses, than the 2 plus. So this guy wins out. He gets the stronger signal for proliferation. This process of just tweaking their DNA ever so slightly, mutating it so that hopefully they get a better fit, a better binding, is referred to as affinity maturation. So these are little small spontaneous mutations purposely done to hopefully get a better binding to the antigen. So remember, each B cell is unique in its antibody, right? That combination of the heavy chain and the light chain in the variable regions there, and just in making those chains. But they'll continue to mutate ever so slightly once they've been activated. The other thing they'll do is class switching. Remember, on the heavy chain, the part of the constant region makes up the stem, right, that FC region of the antibody that interacts with the immune system. And this is why IgG is so beneficial, right, is because it can interact with just about anything in our immune system. So if something's tagged with an antibody, chances are it's going to be destroyed by our immune system. So especially for our lymph nodes, right, after producing the IgM, which all plasma cells are going to normally secrete when they're first activated, helper T cells can sometimes cause them to switch which class. So in the case of our lymph nodes, for the most part, they're going to switch to IgG. Right, this is internally in the body. This can go into the lymph, into the bloodstream. And these are the most protective antibodies we produce. But B cells that are associated with our MALT, our mucus-associated lymphoid tissue, these guys, of course, are going to stimulate those B cells in that area to produce IgA and to produce the secretory protein that's going to hook the two together. Because remember, it forms a dimer when we secrete it. So how do they switch? They literally have to mutate their DNA. So once a B cell switches, it's not going to switch again. Okay, it's a one-time thing. So notice that this particular one, right, is producing IgM, and it has the ability to go to D, G, E, or A. Here, it literally cuts out the M and the D to get to the G to produce G. If it's in the MALT, in the mucus-associated lymphoid tissue, right, it's going to cut out M, D, G, E, and it's going to make A. And it's going to be stuck. That B cell is going to be stuck. Once it makes that change, once it makes that switch, it only produces IgA. It can't switch again. Does that make sense? So the same is true for IgE, right? There are certain individuals, like myself, that are programmed a particular way under certain circumstances. Unfortunately, we produce IgE. As we'll talk about in disorders next, the problem with that particular type of antibody is the cells that it interacts with, right? And the type of response that you get. It's an inflammatory response, which we get most of the time anyways with antibody activation, right? Of course, not with IgA, because that's leaving our body, right? That's secreted. So as we said, right, that second exposure, that activation is really due to memory cells. That's what's helping us out. That's what gets rid of that lag period of where we're searching for the naive cell when we first are exposed to something for the very first time. So there's that animation that we watched a moment ago. So T, thank you. He's not going to be able to hear by the time he's 30. He has earbuds in, and you can hear his music. Unbelievable. So T-cell, helper T-cells are sometimes needed, but B-cell receptors can bind with antigen, right? So that's what you see happening here. And this is one of those situations, especially with lipopolysaccharide, because it's kind of repetitive. It can bind with lots of antibodies on the surface, that B-cell receptor on the surface of the B-cell. So polysaccharides, repetitive, right, lipopolysaccharides tend to be what we call T-independent antigens. So just the interaction of the antigen and some other signaling is needed for the B-cell to produce plasma cells and memory cells. You don't need the helper T-cell to interact. So hence T-independent antigens. So what markers are found on all our nucleated cells and antigen-presenting cells? MHC, class 1 on all nucleated cells, and antigen-presenting cells, MHC class 2. What's the important relationship between the T-cell receptor and the CD markers? This helps them communicate with MHC, right? So we need to know which MHC goes with which CD4. Dendritic cells are very important to this process of presenting to T cells, right? They're one of the main ones that are going to share this information that they've picked up, these antigens. So we have our MHC class 1, and this is mainly endogenous antigens, right? So this is on all of our cells, even our antigen-presenting cells, right? All nucleated cells have this. MHC class 2 is for exogenous antigens, so stuff that's outside, right, that we pick up. And so this is mainly the job of our antigen-presenting cells. And this is why they're the only ones that have MHC class 2. So remember, who are our antigen-presenting cells? Who's going to gobble it up and then present it? Dendritic cells, macrophages, and B cells. Right? B cells need this for their activation by T cells sometimes. And for macrophages, since they're some of our main important killers, sometimes even they get activated by the helper T cell. Mostly, though, your dendritic cells are activating your T cells. All right, so they're a very crucial part of our immune response, going from innate to adaptive, right, where we're communicating with these helper T cells to help out the B cells, right, also communicating with cytotoxic T cells and activating them. So again, all cells, all nucleated cells present MHC class 1, and this is so that if anything that gets inside of us, we can present it. So remember, what type of T cell do we want to present to? Helper T cell or cytotoxic T cell? Cytotoxic. So in this case, a CD8 T cell, right, is going to communicate with MHC class 1. And it's usually going to lead to the destruction of the cell that's presenting MHC class 1. Now, macrophages and dendritic cells can present using MHC class 1 as well. And that's going to activate those cytotoxic T cells. So they'll proliferate, we'll get even more, and they can hunt out the cells that are presenting with MHC class 1 that are infected and destroy them. Where helper T cells are going to be activated by MHC class 2, and they help out other cells, right? So they're going to help the B cell in activation. They're going to help the macrophage in activation. The dendritic cells are going to activate those helper T cells. So that process of antigen presentation.
Depending on whether they originate within or outside the cell. Proteins produced within the cell, such as viruses or cell proteins, are broken down into fragments. Fragments of foreign proteins are antigens. The antigens are then transported into the rough endoplasmic reticulum. The antigens combine with class 1 MHC molecules. The combination is then transported to the Golgi apparatus and from there to the plasma membrane. Foreign antigens presented on class 1 MHC molecules stimulate cell destruction by activated T cells. Self-antigens are not recognized by the T cells and do not stimulate cell destruction. A different process occurs if the antigen originates outside the cell. Phagocytes, such as macrophages, ingest foreign particles, such as viruses and bacteria, by endocytosis. The foreign particles are broken down into fragments within a vesicle. The vesicle containing the foreign fragments fuses with vesicles from the Golgi apparatus containing the class 2 MHCs, and the two structures combine. The MHC class 2 antigen complex is transported to the plasma membrane. The displayed MHC class 2 antigen complex can stimulate other immune system cells to respond to the antigen.
So they're going to communicate with those helper T cells, right? That will help out the macrophage, they'll help out the B cell. As their name implies, they're helpers. So helper T cell always, should be synonymous with CD4 for you now. And the important thing about that is that it can communicate with MHC class 2, which is found on your antigen presenting cells, right? Your B cells, your macrophages, your dendritic cells. Right? It's going to help B cells and macrophages, but typically it's going to be activated by your dendritic cells. Same can be true for your cytotoxic T cells, but their targets, right, those CD8 cells, are any nucleated cells that are presenting that antigen, and they're going to induce that cell to die. And it's going to be programmed cell death, right, apoptosis. So we have protection against stuff that gets inside of our cells, and we have protection for outside. So this is a cellular response when we have cytotoxic T cells. When we get activation of B cells and the production of antibody, remember that's our humoral response, antibody production. And a lot of times, depending, right, we might even get both, especially with viruses, right? They get inside of our cells, but they are outside of our cells as well. So antibodies are beneficial when they're outside, and a cytotoxic T cell response is beneficial when it's inside. For pathogens like Mycobacteria leprae that get inside your cells and actually can grow inside your macrophages, it's important to have a cellular response, right, and not just an antibody response. If you go all in one direction, like with lepromatous leprosy, then you run into the problem that you're only producing antibodies, and antibodies can't get into your cells. You need a cellular response when you have intercellular pathogens, right? So a lot of times our body is going to respond both ways, both antibody and cellular. But if we respond only in one way, then we're not always going to eliminate the pathogen, depending on where it goes. Does it stay outside of our cells? Does it get inside of our cells? Right? Antibodies are limited to outside of our cells, where our cellular response, those cytotoxic T cells, can detect, as you can see, when cells are presenting antigen. Your cells are always presenting antigen. They're always taking pieces from inside, as you saw, and presenting it on the outside via MHC class 1. So they can always communicate with our cytotoxic T cells. So we're always checking. Right? So B cells have the B cell receptor, which can directly interact with antigen, where T cells are going to have their T cell receptor. And again, just like the B cell, they make different receptors, different variable regions. You as a person make specific MHC molecules as well that can bind to the antigen, that vary. So this one just includes some additional pictures, right? So notice two things on the T cell. If it's CD8, we know that it's a cytotoxic T cell, right? It can talk to any cell presenting MHC class, any one of our nucleated cells. Where helper T cells have CD4, they're only communicating with cells that have MHC class 2. So they're only communicating with dendritic cells, B cells, and macrophages. Whether it is to activate that helper T cell, which is commonly done by the dendritic cells, or to activate the B cell that they're communicating with, or activate the macrophage they're communicating with to become a better killer. So as I said, dendritic cells are the main ones that are important for this activation. And they can present both MHC class 1 and 2. So they can activate cytotoxic T cells as well as CD4 helper T cells. Just depends on what type of antigen they've gobbled up and what type of response we need. And we could get both happening, right? That isn't the only thing that happens. Notice that additional signaling is necessary, but we're going to keep it at this basic level. Remember I said we were actually going to just be scratching the surface? I know this seems like a lot already, and it actually goes into more detail than we're covering in this course. But this is the level that you need for this level. So we'll continue on this next time, but again, this important ability to detect and kill, right, by those cytotoxic T cells. Make sure, right, especially if you came in late today, watch those videos that I have of an activity I've done with my class in the past. Write about 10 different points that you remember from those videos to get your 10 bonus points by Wednesday.